So good evening everyone. Today we're talking about sheath blight of rice. So basically sheath blight of rice is considered to be an important disease next to rice blast. Rice sheath blight is an increasing concern for rice productions, especially intensified production systems in Japan. The disease causes highest yield loss like 50% within the susceptible variety, and in basically it's losses the yield like up to 20 to 25%. Let's talking about causal organisms. Basically, seed blight of rice caused by Rhizoctonia solani. It is an soil-borne pathogens, and these pathogens grow well on common culture media. The fungus produce septate mycelia, which is are hyaline when it is young. When it's become old, the color will be changed like yellowish brown. It's produce large number of spherical brown sclerosia on the media, and the fungus grow over a wide range of temperature. So asexual reproductions. Rhizoctonia solani does not produce any asexual spore or conidia. The fungus infected plant by vegetative mycelia or germinated sclerosia producing hyphal thread. The mycelia produced long multinucleate cells that grow approximately at the right angle to the main hyphae, with a slight constructions like the junctions of main hyphae to branch. And the hyphal cells are septated by the septum containing as a pore which is allowed to movement the mitochondria, nuclei, and cytoplasms from the cell to cell. Let's talking about symptoms. The fungi affected the crop from tillering to heading stage. Initially, symptoms are noticed on leaf sheath near to water level. On the leaf sheath, oval or elliptical or irregular greenish gray spot are formed. As the spot enlarge, the center becomes grayish white with an irregular black, brown or purple brown border. The lesions of upper parts of plant extend rapidly, coalescing with each other and cover the entire tillers from the waterline to flaglets. The presence of several lesions or leaf sheaths usually occur death of the whole leaves. In severe cases, all the leaves of the plant may be blighted. Older plant are highly susceptible, plant highly infected in early headening and grain filling stage, and poorly filling the grain is the ultimate result. Especially like the lower part of the panicles is more infected. So if we're talking about the epidemiology or most suitable temperature or humid conditions, like the temperatures required for this fungi growth like 25 to 30 degrees Celsius, more than 95% humidity is most preferable for this fungi, and highly like a heavy dose of nitrogen fertilizers make the tissue more susceptible, and like while high rate of potassium induce the resistance to the disease. Let's talking about disease cycle. Basically, the sclerosia and mycelia of infected debris is a primary source of inoculum. During the cropping seasons or at the harvesting stage, the sclerosia fall under the ground and serve as the survival structures of this fungi. And basically, the sclerosia is survived up to 2 years in next cropping seasons, and the survival is more frequently accumulated to rise over the time. The field water movement and irrigations are the main source of dispersal of sclerosia and the infected crop debris. Basically, the initial infection started from the sclerosia when it is germinated and it's come contact to the susceptible host plant, and it may germinate through natural opening or in by means of specialized infection structures called the appressoriums. And the fungus also produce extracellular enzyme that degrade the plant cells and facilitate the colonizations, or one of the fungi penetrate to colonized and the plant tissue, and it will show the symptoms, and rapidly it may attack to healthy plants and it become a severe, and that can causes high yield losses. Talking about management, the cultural management, we can grow the resistant variety, the Swarnadan, Radha, Pankaj, and Madhya Vijaya, and we can avoid the infected seed, uh, and we can use the healthy seed, apply the seed dose of nitrogen fertilizers, and check the brown plant of our populations and destroy the stubble of weeds. Biological management, we can apply Pseudomonas fluorescens 2.5 kg per hectare after 30 days of transplanting. We can mix 50 kg of FOM and apply it in the soil. As in foliar spray, Pseudomonas fluorescens you can use to 0.2% at the boating stage and 10 days later, and apply the organic amendment like neem cake 150 kg per hectare or FIM 12.5 ton per hectare. In chemical management, we can use first seed treatment with Thyram 75 WP 2.5 gram per kg seed, and we can apply the foliar spray with hexaconazole contact 5 EC 2 ml per liter, propinaconazole till 25 EC 2 ml per liter, and carbendazim 2 gram per liter as a 10 to 12 day interval. Thank you, everyone.